let us continue with the liquid liquid extraction. Generally in liquid liquid extraction we use a solvent to extract the desired solute, but there are situations where you would like to use water instead of an organic solvent. For example, if you want to recover proteins, proteins may denature in organic solvent, so you would like to use water. There may be situations where you may like to transfer from one water phase to another water phase, so again you are talking about aqueous medium. So there are certain different strategies where you adopt when water is also present, either as a solvent or as both the heavies as well as the lights. One of the important liquid liquid extraction technique is called the reverse micellar extraction. Let us look at what is a micelle. Okay. Before that we will look at what is a surfactant or a surface active agent. Surface active agent, it always stays at the interface because you have a water layer and you have a solvent layer, so the interface is between the water and the solvent layer. The surfactant has a polar head group. A polar head group could be a OH, or it could be a nitrogen, or it could be some sort of an ion, and then it will have a hydrophobic tail. So hydrophobic tail could be a hydrocarbon type of a small C8 hydrocarbon, it could be C12, it could be even C18. There are surfactants which have very very long hydrophobic tails. So hydrophobic groups like prefer solvent layer, and the hydrophilic or the polar groups prefer the water layer. So if you have an interface, that means if you have an organic solvent and a water and you put in surfactant, the polar head group will always go towards the water layer, whereas the hydrophobic tail, as we call it because it is a long chain hydrocarbon, we call it a tail, this will always go to the organic layer. That is how a surfactant partitions at the interface. Now what happens, suppose I have a water and protein and there is surfactant. Now I add a solvent, what will happen? The surfactants will spontaneously aggregate in the organic solvent to form something called a reverse micelle. What is a reverse micelle? If you look here, you have all the polar head groups inside, that is they are away from the solvent which is outside, and all the hydrocarbon portion is in the solvent region. So inside region is like a small cage which is highly hydrophilic, that means there will be water inside, and if there are any water soluble solutes, proteins, all of them will be inside because it is a hydrophilic region inside, outside is hydrophobic. That is why this is called reverse micelle. Here the polar head groups are inside, buried inside, whereas the hydrocarbon chains are outside in the solvent layer. So this is what is called a reverse micelle. So where do you use reverse micelle? Reverse micelles are very very important. If we have a protein which is water soluble and we want to barrier it, or we want it to be kept away from a organic solvent, because organic solvents may denature your protein. So you want to keep it away from an organic solvent, then we may use a concept of reverse micelle. Now what happens, you have the solvent outside, you have the surfactant which is forming a reverse micelle, you have a polar region inside where you may have water, you may have buffer, that means salts present, and your protein also present inside. So that is what is a reverse micelle. So we can use this concept of reverse micelle in extraction, especially for solvent sensitive proteins, that means proteins which may get denatured because of the solvent. So we have, as I said inside before, we have the organic solvent, we have the reverse surfactant aggregates, the polar head groups are located inside and the hydrophobic tails are pointing outside. So inside you may have a core which is aqueous, so the protein can be kept inside. So you are protecting your protein. So you can have many surfactant aggregates like that present inside the organic solvent, and each surfactant aggregate will have the required protein. So all you need to do is remove the aggregate from the solvent and then you make the surfactant leach out the desired protein. So this is how you recover proteins. Now what are the factors that affect? As you can realize, it is an aqueous system, that is where the reverse micelles are formed. So there are many factors which affect reverse micelle. Most important thing is the pH, the ionic strength, the type of salt we use, type of organic solvent, type of surfactants we use. So there could be surfactants which will form a very good reverse micelle inner core, so they may prevent the protein leaching out from the inner core. So there is going to be a lot of electrostatic interaction between the protein and the surfactant polar head groups, which solubilizes the protein in the reverse micelle inside. So if you have very strong forces between the protein and the surfactant, you are going to dissolve more of the protein in the aqueous inner core. So by selecting these conditions, especially the type of surfactants you take, the type of salt you add, the changes in pH as well as the ionic strength, we will be able to solubilize more of the protein. That way we will be able to extract things better. So if you have you all know what is an isoelectric pH. That means isoelectric pH is the pH at which a protein will have net 0 charge. So if you know what is the isoelectric pH of a protein, and if you are operating at a pH less than the isoelectric point of the protein, so the solubility of the protein at pH less than the isoelectric pH is greater in an anionic surfactant, and at pH greater than the isoelectric pH of the protein, it would inhibit reverse micelle extraction. Okay. So if the pH is greater than the isoelectric pH, then you are going to prevent the reverse micelle extraction. So solubility of the protein would be more at pH greater than the isoelectric pH if the protein has a net negative charge in a reverse micelle formed by a cationic surfactant. Okay. So depending upon the pH, we can select the type of surfactant. Do I need an anionic surfactant or do I need a cationic surfactant? Okay. So if you want to work at a pH which is greater than the isoelectric pH of the protein, then ideally I should have a cationic surfactant. Okay. Otherwise if I am going to work at lower pH, lower than its isoelectric pH, then it will be good to have a anionic surfactant. So by selecting the surfactant depending upon the pH at which you are going to operate, you can increase the solubility of the protein. Or conversely, if I know what type of surfactant I am going to use, then I will select the pH either greater than the isoelectric pH or less than the isoelectric pH. By adjusting the pH based on the isoelectric point, I can increase the solubility of the protein in that particular surfactant. So you see there is a very close interaction between the type of surfactant I select, anionic or cationic, and the pH at which I operate with respect to the isoelectric pH. So there are many conditions by choosing we can alter the solubility of the protein in a reverse micelle extraction. So if I have higher salt concentrations, then it is going to screen the electrostatic interaction. That means the electrostatic interaction between the protein and the polar head group is going to be barriered or screened. So I am going to decrease the protein solubilization. So if the salt concentration is increased, then if there is going to be very strong electrostatic interaction between the protein and the polar head group, it is going to get weakened. Similarly, if I have a higher ionic strength, this results in the formation of small reverse micelle. So when I am forming very small reverse micelle, then my solubilization of the protein is reduced because of the size of the core of the reverse micelle hydrophilic region. Whereas if I have a lower ionic strength, then I am able to make larger reverse micelle, that means I will be able to solubilize more of the protein because of the size exclusion effect. Okay. Increased ionic strength, of course it brings out salting out of the protein from the micellar phase. So if I am going to have a very high ionic strength, you know what is the salting out, the solubility of these ions will reduce the solubility of the protein. So the protein solubilization decreases actually. Increase in the surfactant concentration is going to increase the formation of reverse micelles. So I am going to increase the solubilization of the protein, but of course you need to consider the economics of the entire process. Do I need to put in lot of surfactant, or can I afford to waste lot of surfactant? So you need to consider all these points actually. So if you have very high electrostatic interaction between the protein and the hydrophilic head group or polar head group, if I put salts, I am going to create a barrier, number one. If I am going to have very high ionic strength, then the size of the reverse micelle is going to go down, so solubilization of the protein also goes down. If I have too much salt, then I may be creating a salting out effect. So you need to be very careful about the whole thing. So if I have reverse micelle formed by ionic surfactants, they are small to accommodate large proteins, while non-ionic surfactants, they will form large micelles to dissolve a range of proteins. So now this point is very important if you are comparing an ionic surfactant versus non-ionic surfactant. So if I use a non-ionic surfactant, I may be able to form larger micelle when compared to ionic surfactant which may form a smaller micelle. So obviously as I said before, when you have very small core of reverse micelle, the amount of protein that is dissolved also goes down. In addition to reverse micelle, I may be able to incorporate something called affinity partitioning. So I can have a polar head group of the surfactant which is a inhibitor for a particular enzyme. So that particular enzyme may be selectively getting trapped inside the aqueous core. So this is a very intelligent way of improving the selectivity of the protein which you are extracting. So if the polar head group, if I have certain inhibitors attached to the polar head groups, then the enzymes or proteins may be selectively captured inside the inner core. So that is called affinity partitioning in reverse micellar extraction. So typically that is what happens, you have the aqueous phase, you have a protein. So when I put in organic phase, what happens, the reverse micelle is formed. So all the hydrophobic tails are pointing out, hydrophilic polar groups are inside buried, and your protein also is inside buried. So now I have lots of micelles with just the protein, so I can nicely separate them. So it is a very intelligent way of separating proteins. As I said, suppose I have water in which I have certain proteins which I like to extract, again with water itself, because as I said before, proteins are very sensitive in solvent environment and they may get denatured. So you would like to use water itself as an extracting medium. So how do you do that? So we add something called a biopolymer. So the biopolymer in aqueous medium may interact with the protein of our interest. These are called non-bonded interactions. So the interactions could be because of charge, it could be hydrogen bonding, it could be van der Waal interaction between the solute molecule or protein and the polymer molecules in the liquid phase. So what happens? So I may have a system where a protein is present in an aqueous medium, I add another aqueous medium as a solvent where I may have a biopolymer. So there could be an interaction between the biopolymer and the protein gets extracted into the solvent, which is again a water actually. So what are the parameters that influence in this particular situation? The molecular weight of protein. So I am going to show you some interesting results where molecular weight of the protein affects the extraction efficiency very drastically. Mostly we use biopolymers like PEG, dextran, chitosan. So all these are biopolymers and they will not have any adverse effect on the protein. So that means the protein will not get denatured by using these biopolymers. The type of concentration of salt we use, and of course the pH and the temperature, all these factors affect this aqueous two phase extraction system. So the partition coefficient of the protein into the water containing the biopolymer will be function of many parameters. As I said, they are all non-bounded interactions. So the interactions could be electrostatic in nature, it could be hydrophobic in nature, it could be hydrophilic in nature, conformational, ligand binding and so on. So you could have a large combination of partition coefficients all getting added up to give you the overall partition coefficient for the protein. So typically, so if you are talking about a cell fragments, DNA, the K value will be of the order of 100. If it is protein and enzyme, the K value will be of the order of 10. The type of polymer which you use as well as the molecular weight of the polymer which you use for extracting the protein will have very strong effect on the extraction efficiency. This particular example tells you the molecular weight of PEG is affecting the extraction very dramatically. For example, you are trying to extraction of fumarase from a protein mixture using PEG potassium phosphate system. So potassium phosphate is your buffer, PEG is the biopolymer which you are using, okay. So the K value increases, that is K is the partition coefficient value increases with increasing the molecular weight of PEG. So if when I change from 400 as I keep on moving right up to 4000. So increasing molecular weight of polymer in one phase decreases the partition of the protein in that phase. So if I move from 400 to 4000 in one phase, thus the partitioning of the protein in that particular phase decreases. So all I have to do is I keep on adding higher molecular weight PEG. So the solubility of the protein in that phase decreases and it moves to the another phase. This is how you enhance the partition or separation of the protein from one phase to another. Another example, suppose you want to partition pullalanase, this is a enzyme in PEG phase containing PEG dextran. So the K values decreases from 1.3 to 0.25 as the molecular weight of the dextran is increased from 1500 to 6000. So at higher PEG value or higher PEG molecular weight, the K value decreases. So that means I can reduce the solubility of the enzyme in that phase by adding higher molecular weight biopolymer. So why does that happen? It is almost like salting out effect. So you are adding a larger molecular weight material, so the protein leaves the system and goes to the another phase. So what type of equipments do you use in two phase extraction, aqueous two phase extraction? We can use batch, we can use continuous, just like we are using solvent based systems. So continuous has a lot of advantages when compared to batch, but the advantage of batch is you just use a very simple tank, you mix this two systems and let the two phases to separate, and then you just do separate it through gravity, whereas in a continuous system you need to have many equipments, many controls and so on. But then their advantages are tremendous. You get better product uniformity, you get better purity, you can go into automated operation, we have better control of partitioning conditions. So that is the one of the main advantages. We can even go for smaller process equipment when compared to batch. Batch you may require a very large tank, whereas in a continuous system we may do the whole job in 1/4 or 1/5 the size of this large tank, and it is easier to integrate with the downstream processing. So I may have a continuous liquid-liquid extraction, then I may take it to a chromatography which may be operating continuously, I may go to a filter which may be operating continuously and so on. So we can nicely integrate continuous operation, whereas batch operation you are going to have a product and for some time there will not be any product coming out, and then again there will be a product and so on. So it is going to be start stop start stop. So that will be the main difference between a batch and a continuous operation. So we need to think about large scale operations, because whatever is done in the lab needs to be scaled up and taken up to kilograms, kiloliters, meter cubes and so on. So very large scale operations. So when we talk about very large scale operations, there are couple of very important parameters we need to consider. One is the viscosity, other is the interfacial tension. So if you are handling very highly viscous material, for example fermentation broth is going to be very highly viscous, because you are going to have dead cells, you are going to have cell debris, you are going to have intracellular material, you are going to have DNA, so many components. So the viscosity is going to be highly non-Newtonian, and the values are also going to be very high. So when you are going to have very highly viscous material, now you are adding a solvent to extract a protein or a metabolite. Because of the high viscosity, there are going to be mass transfer limitations both during the extraction as well as in the phase separation, because after you do the extraction you are going to allow both the phases to separate, because the phases are highly viscous, the separation also is going to take a very long time. Not only the extraction is going to be mass transfer limited, the separation is also going to take a much longer time. That is one problem. Problem number 2, if you are going to have low interfacial tension in aqueous two phase system, you can have small emulsion droplets. So when you are going to have emulsion droplets, because you are having water, you are having solvent, you are having surfactant, and interfacial tension is also very low. So what happens, you are going to have small emulsion droplets. So the phase separation is not going to be very clear. You are going to have a liquid aqueous phase, you are going to have a liquid solvent phase, you may have in between some emulsions formed, the emulsions may not separate out clearly, so you may lose some of your product in the emulsion and so on actually. So low interfacial tension has this type of emulsion formation and it will affect the separation efficiency. So all these two important points we need to consider when you are scaling up this type of aqueous two phase systems. So people have considered inline and static mixers, that means you will have long tubes filled with the certain static elements, and when the two liquids flow through it, they get mixed, and hence during the mixing process extraction takes place. This type of static mixers have been considered. Once you mix them, you use centrifugal separators, different types of centrifuges. We talked about different types of centrifuges. You can use disc stack centrifuge, you can use nozzle separators or decant extractors and so on. So you generally in a two phase extraction where you are using aqueous as one of the phases, and if the viscosities are very high or if the interfacial tension is very low, you go for a inline mixing followed by a centrifugal separator, and nowadays you get very good liquid centrifugal separators. That means they can separate two liquid phases very nicely, just like they can separate out a solid and a liquid. We can use both batch as well as continuous type of operation for a aqueous phase system. Where do you use aqueous phase extraction systems? I did tell that when you are handling enzymes or proteins, they are going to get denatured, so you do not like to use solvents, and in such situations you will go for aqueous systems. So if you are requiring enzymes like fumarase, penicillin acylase, formate dehydrogenases, all these enzymes from fermentation broth, fumarase recovered from baker's yeast, penicillin acylase from recombinant E. coli strain. So these are some situations where you will use aqueous systems. If you are recovering bulk protein, especially valuable proteins, you don't want the protein to get denatured because it is a very valuable protein, then you may go for aqueous extraction systems like alkaline protease for licheniformis by aqueous 2 phase extraction using PEG and Rapal 200. Rapal is nothing but a partly hydrolyzed hydroxypropyl derivative of starch. That means you are using a biopolymer, both are biocompatible, so the protein does not get denatured during the extraction process. You can use this type of aqueous system for partitioning of cell debris as well. So once I am disrupting a biomass because my product is intracellular, so when I disrupt a biomass you are going to have desired product, you are going to have viscous colloidal suspension, organelle, cell wall fragments, other particulate matter, salts and so on. General solid liquid separation may be difficult. That means solid liquid separation, if you recall, we use a filter or we use a centrifuge, that may be difficult. So here we can achieve this separation by adding a aqueous system containing PEG, potassium phosphate extractant. So this will partition the cell debris and particulates into the heavier raffinate phase. So by adding this aqueous biopolymer with salt, I will be able to separate all the cell debris into the heavier raffinate phase. So that way I will be able to recover my intracellular desired product. Affinity partition is it's based on molecular recognition. It is like affinity chromatography. We will talk about chromatography later. So you have a ligand which has an affinity for a protein. As you know, a ligand protein interaction is very selective. So if I have a ligand which is going to bind to a protein very selectively, then I will be able to extract only that protein from a mixture of large number of proteins. So that is what is called affinity partitioning. So I need to know what is the affinity ligand which is needed to selectively bind to the protein, and then I will have that particular ligand present in my solvent. So the protein will just selectively bind to the ligand, thereby I can separate out. So how do you keep the ligands in your solution? So one of the polymers which you are using, a biopolymer, you can have the ligand covalently bound to the biopolymer, so that you can later on reuse the whole system again and again. So if I have a affinity ligand, so if the protein has to be extracted and it has n number of independent binding sites for the affinity ligand, then the partition coefficient K in the presence of the affinity ligand will be like this: K equal to K naught, where K naught is the partition coefficient of the protein in the absence of ligand. So if I do not use the affinity ligand concept, then my partition coefficient will be K naught, but because I am adding an affinity ligand which may bind to the protein at n different locations, then the K, the partition coefficient, gets modified like K naught into 1 plus L divided by KT raised to the power n divided by 1 plus L divided by KB and KL. What is L here? L is the total ligand concentration. So as you can see, if I have more ligand present, obviously my K will increase like this. Okay. Now what is KB, KT and KL? Now KL is the partitioning coefficient of the free ligand. As you can understand, the ligand also can get partitioned between the two phases. So some amount of ligand is going to get lost because of the partition coefficient, and then KB and KT are the dissociation constant of the protein ligand complex in the bottom and the top phases. So now you have a protein ligand complex which may be present in the bottom, that means the heavies or the raffinate, or it may be present in the lights or the top or the solvent phase. So it can dissociate in this phase, that is the heavy phase, or it can dissociate in the light phase, or it can dissociate in the bottoms, or it can be dissociated in the top, and the dissociation constants may be different. So that is why you have KB dissociation constant in the bottom, KT dissociation constant in the top. So, you see the partition coefficient for the protein varies and it depends on the concentration of the ligand I am using, the number of sites available in the protein for the affinity ligand to bind, the partition coefficient of your ligand, and the dissociation constant of the protein ligand in the heavy phase as well as in the light phase. So, I can select a suitable ligand so that I can enhance my partition coefficients tremendously. I will be able to enhance the extraction efficiency by a very large number by selecting suitable ligands and also by selecting suitable ligand concentration. So, the binding constant of the for the protein ligand complex determines the selectivity of the extraction. So, the stronger the binding of protein to the ligand, the lower the ligand concentration required for selective extraction. That means if it is going to bind very strongly, I can use less ligand amount. And also the selectivity increases exponentially depending upon the number of sites present. If the number of sites because the term n comes as an exponent, that is why the selectivity increases depending upon the number of sites in an exponential manner. And also the number of binding sites determines the yield. So, if I have more binding sites, I am going to increase the yield of extraction. So, what are the advantages of affinity extraction? We can use it for extracting different enzymes from yeast porcine muscle. We can extract formate dehydrogenase from a cell of Candida boydini and beta interferon from a mammalian cell culture medium and so on actually. So, we can very selectively extract a protein from a mixtures of a large number of protein. So, the extraction efficiency, the selectivity or the efficiency is going to be affected by many factors. The nature of affinity ligand I am using, the phase forming polymer, because your affinity ligand is going to be immobilized on a polymer. We can't just have the free affinity ligand floating around. So, it is going to be on a polymer in a covalent manner. So, the phase forming behavior of the polymer, the temperature at which you are studying, the pH, the protein concentration, the type of salts used and the concentration of the salt. So, these are some of these are very standard parameters which are going to affect in the aqueous system. For the affinity, the nature of the affinity ligand and the nature of the polymers are two different parameters which needs to be considered as well while selecting a ligand. Now, let us go to the limitations of liquid-liquid extraction process. We have looked at a large number of extraction approaches. We have looked at mathematical relationships, we have looked at aqueous systems both as just the solvent, we have looked at the reverse phase approach, we have looked at the affinity approach and so on. So, there are many limitations of the liquid-liquid extraction, it is not 100% the best. We have to find a suitable solvent. So, that is one of the headaches. You want the solvent partially soluble with the feed, you want the feed components immiscible with the solvent, that means we do not want other components to be dissolving in the solvent. So, solute is soluble in the carrier, at the same time it should be completely soluble in the solvent, then only the solute can partition from the carrier into the solvent, that means carrier is nothing but the heavies. Okay. If the density differences are not much, then you are not going to get two distinct clear phases. So, these are some of the limitations of liquid extraction. So, what happens if the solvent is partially soluble in the feed, you are going to lose some solvent. If the feed components are not miscible with the solvent, you are not going to get a good mass transfer and movement of the feed components into the solvent. Okay. So, if the solute is soluble more in the carrier than soluble in the solvent, the solute partition is also going to be a big problem. So, these are limitations with respect to the solvent. Now, the K value has to be sufficiently large. K is nothing but their partition coefficient. So, if the K value is large, then you will have less number of theoretical plates or theoretical stages. If the K value is small, I need to keep on doing the extraction many times. We have looked at many problems, both the counter current, co-current, cross current systems, and there you have realized if the K value is very small, I need to keep on contacting both the solvent layer and the heavy layer in many stages to achieve a efficient separation. Viscosity, if the viscosity is very large, then your settling is going to take much longer time. So, I should have low viscosity. Now, the solvent must be chemically stable. You do not want a solvent which is not chemically stable, it may start reacting with some of the solute molecules or feed molecules. So, it should be inert totally with respect to other components. It should be cheap, should not be toxic, because we are talking in terms of using large amount of solvent, and it should not be flammable as well. So, that means it should be non-flammable. So far we looked at the solvent. You see, solvent selection is a big challenge and it should satisfy a large number of conditions. Now, let us look at equipment. So, if the interfacial tension and viscosity are high, obviously I need to put in more power to achieve a good mass transfer. So, if I am mixing two phases and the viscosity is high, I will put them together and I will have an agitator, and the agitator has to revolve faster. So, the power required also goes up. So, higher the viscosity, higher will be the power requirement. If I am having low interfacial tension, it will also lead to formation of emulsion. So, I need to be very careful that if I agitate too much in a low interfacial tension system, I will be forming small emulsion droplets, and that means I am losing my product in those emulsions, and breaking the emulsions is not so feasible. Temperature, if I increase the temperature, solubility of the phases also increases, but then I do not want to increase the temperature too much that it is coming closer to the critical solution temperature. Pressure, I should maintain the pressure below the vapor pressure of the solution, otherwise I do not want the solvent becoming vapor or some of the feed components becoming vapor. This happens if you are using very low boiling solvent. If I am using say pentane, or if I am using a very low boiling hydrocarbon, you may have the hydrocarbon both in the liquid and the vapor phase. So, some of the hydrocarbon will be lost, as well as you are thinking about in terms of the flammability issues also. So, pressure is also very important if you are dealing with low boiling solvents. So, how do you select the extractor? So, the extractor selection depends upon the concept of mass transfer. So, obviously you want to increase the surface area between the two phases, that is the solvent phase and the heavy phases. That is one thing. Other one is the flow of the feeds should be adjusted so that you get maximum solute recovery. That means as much of the solute that is present in your medium is recovered, and if it is a protein which is high value, then you want to recover almost 100% of the protein into your solvent layer. So, these are the two factors which affect your selection of the extractor. We talked about broadly three types of extractors, if you very clearly remember. One is the mixer settler, the other one is the column type, third one is the centrifugal. So, different types of columns we looked at, different types of mixer settlers, different types of centrifuges. So, each mixer settler is almost like one equilibrium stage. So, if I am interested in only one mixing then I may go for a mixer settler, but if I am interested in 4 or 5 different stages, then it is not very efficient to have 4 or 5 different mixer settler, then you may go to some other type of design. Why? Because they will occupy lot of space on the ground and they will also require large volume. The vessel size has to be very big. So, if the vessel size are big, then you will require high liquid demand, that means I may have to put in more solvents and so on. Actually, columns are tall structures where you are bringing both your solvent and the heavies either in co-current or counter current fashion, they get mixed thoroughly because you will have trays or you will have packing inside the column. So, during those mixing stages inside the column, the partition of the solute takes place. So, columns are very good, they occupy less space. I can have many stages, I can put in many separators inside the column in the form of a tray, and each portion is almost like a equivalent to a stage, each portion is almost like an ideal stage. So, it is very good for liquid extraction. So, I can use packings, I can use trays, thereby I can increase the surface area of contact between the two liquids, so that the two liquids intermingle nicely. So, by putting in packings, I am increasing the flow path of the solution. So, the solution will travel through the interstices or spaces in the packing. So, they are coming in contact with the other liquid. So, I am increasing the flow path. But one important point is the packing material should be wetted by the continuous phase. So, you cannot have very low flow rate that the packing is not wetted at all, there are some dry regions, then you are not efficiently making use of the packing. Generally counter current is preferred because you get better separations or better efficiencies, and of course as I said it occupies less space as well. The third type is the centrifugal contractors, where you have liquid centrifuges which can separate very close compounds or solutions which have very close in density, that means almost a difference of only 4% density difference can be separated. And also if it requires many equilibrium stages, centrifugal separators are very good. We can operate nicely in continuous mode, it occupies less space as well. So, 3 different types of equipments, each one has got advantages, disadvantages, and nowadays the general public or general industries move towards either a column type of approach or a centrifugal contactor, unless they just want to do one single extraction process where they may go for a mixer settler. Each one of them have different cost depending upon the type of equipment we do, depending upon the type of number of stages, the cost of the purchase of the equipment, installing the equipment, maintaining the equipment varies. So, I am going to give little bit idea about the cost factors also, because in downstream processing it is not only designing the process, but one needs to get some feel about the cost as well. Okay. The cost data was taken from a book called Liquid-Liquid Extraction CH396 and a large number of authors. So, at a fixed solvent to feed ratio, the amount of solute extracted increases the increased number of trays. So, obviously, so I am fixing the solvent to feed ratio and I want to keep on extracting. So, if I have many stages, I will be able to increase the amount of solute that is extracted. The value of the unextracted solute must be balanced against the cost of the extraction equipment, but then I can put many stages, but then I am going to increase the cost of the overall liquid extraction process. Is it really worth extracting all the solute? So, it depends upon the price of the solute, is it a very value added chemical or is it a bulk chemical. So, if I have a very cheap solute which I need to extract, there is no point in adding extra stage or another extra liquid extraction system to extract as much as possible, because the cost of the equipment may be much higher than the amount that is being extracted. So, you need to balance that, whereas if it is a pharmaceutical enzyme, you would like to extract as much of the enzyme as possible because it is extremely expensive, then you may put in one more liquid extraction stage so that you extract as much as possible. So, you need to determine, should I increase the number of stages or am I happy with the less efficient extraction itself. For a fixed extraction efficiency, the number of stages required decreases as the solvent amount increases. So, suppose I have 10 extraction stages, then I will require certain amount of solvent. For the same extraction efficiency, if I am going to put in 12 stages, then obviously the amount of solvent required also will go up actually. Okay. So, if the number of stages required decreases, the solvent amount increases. So, if the number of stages increases, the solvent amount decreases. So, this is also very logical and obvious actually. But then if I am going to have less amount of stages and I am going to have more amount of solvent, obviously the size of the stage has to be sufficient for handling so much of the liquid. So, each of the stage may be of very big size. So, you see the advantage of reducing the number of stages is balanced by increasing in the size of the stage. So, when I am going to have a larger sized stage, then the cost increases, but I may be having a less number of stages. So, you need to find out which is more efficient. Is it good to have less number of stages which are very large, or more number of stages which are very small. So, because this type of contradictory forces are happening, the cost of the equipment passes through a minimum. That means you will have minimum number of stages that are utilized actually. Okay. So, you need to balance all these factors so that you decide on how many number of stages. It is not only the stage efficiency. If you are fixing the efficiency, do I increase the solvent amount or do I decrease the solvent amount, and depending upon the larger amount of solvent or lower smaller amount of solvent, the stage size increases or stage size decreases. Another point you need to keep in mind is, if the amount of solvent going to be used is more, obviously solvent recovery, solvent removal, all those operating cost, handling cost also increases. So, that is also adding to the overall utility cost. So, that is an extra point which you need to keep in mind as well. So, I can balance with the number of stages, size of each stage and the solvent amount for a same efficiency of extraction, but all of them will have different cost angle. So, I need to look at different scenarios and find out what the cost of each of these scenario, and then make a decision for a fixed extraction efficiency. So, it is not so simple. For a fixed extraction efficiency, it is not so simple by saying that I will require so many number of stages and so much amount of solvent per hour or per minute, so on actually. Now, let us look at each one of these extractor and what are the various factors that contribute to the cost, and how does the cost factor look like as a function of various parameters. Now, in the very first on the second classes, we talked about capital cost and what are the various factors that are involved in capital cost. Capital cost, it is not only just purchasing the equipment, but also it is installing the equipment. That means you are making the equipment usable. So, these factors of making the equipment usable also need to be considered when you do the capital cost. So, it is a combination of purchase cost plus installation cost. Installation cost involves so many factors, adding pipes, adding electricity, adding motors, adding utilities, making the land, making the foundation, all those things, all these costs add up, and that is how capital cost is calculated. So sometimes I may purchase an equipment for rupees 1 lakh, but by the time I make it operational I may be spending another 2 lakhs. So the actual capital cost will be 1 plus 2 lakhs. So you need to consider the other factors also. So capital cost of a column is made up of many factors: diameter of the column, the height of the column, and the material of construction of the column, what is the pressure you are operating the column at, all these factors actually, and all these factors have an effect on the capital cost based on certain exponents, and that is what these equations tell us, okay, which we will continue in the next class.